Okay, good afternoon. So I'm going to tell you about the new core facility that we established at SEBS, which is on the Cook campus, about 10 minutes from here. So this is the building where our core facility is, which is established in 2011 with funding from NIH. So this is a picture of Foran Hall, and we are in the second floor of this building. Our facility is managed by Mike Pierce, who's also going to tell you about some of the work he's doing. We had our first open house on March 15, 2012, and we have an internal advisory committee that consists of faculty from different campuses. So in the core facility, we have an advanced biological imaging core, which has a Zeiss LSM 710 confocal microscope. And you're going to hear more about this microscope in the talk right after my talk. And this microscope has 7 lasers on it, 6 standard lasers, in addition the 594 for visualizing M-cherry and some red dyes. It has a computer-driven XY stage for looking at different regions of the sample, and has FRET-FRAP software for this kind of analysis. In addition, it has the Image Wizard Plus software that can do 3D or 4D rendering of the images. We have an environmental control chamber, which can allow us to regulate temperature and do time course or motility type experiments. So we've been using this microscope to look at some of our plant samples, and this is an example of a lipid transfer protein expressed in tobacco, showing visualization of the protein in the cell wall. In addition, we have an Olympus FSX 100 microscope, which is an inverted fluorescence phase contrast and bright field microscope with a colored digital camera, which we use it for transfection experiments to figure out the efficiency of transfection. And there is more information about this in our website. So in the advanced imaging core, we also have a Typhoon, which is made by GE Healthcare. And we use this imager for Western blots using fluorescent antibodies. And this allows us to detect more than one protein at the same time by labeling it with different fluorophores. We can do 2D dye gels for looking at differential expression. And we have the software, Decider 2D differential analysis software, for doing the 2D gel applications. So one of the other subcores that we have in the core facility is called Biomolecular Interaction Analysis Core, which consists of surface plasmon resonance instruments. So the first instrument we got is the Biacore 3000, which is currently being shared by the School of Pharmacy on the Bush campus. Subsequently, we got a Biacore T200 from GE, which is in the core facility. So we use these instruments to look at protein-protein interactions. We can also study protein nucleic acid or protein small molecule interactions. And the advantage of these instruments is that we can get kinetic information. We can get the rate constants, on and off rates, and also the overall dissociation constant. This is an example of binding of a toxin, which is shown in the slide, to a ribosome. And the tracing shows the interaction, the kinetics of this interaction. And you're going to hear more about this from Xiaoping, who will tell you about these instruments later on. And we're teaching hands-on courses on this technology, and this also will be explained in Xiaoping's talk later. So in addition to the Biacore instruments, we also have an ITC, which is a MicroCal ITC 200. And this instrument can also measure protein-protein interactions and determine the affinity based on the thermodynamics of these interactions, either based on the heat generated or heat absorbed. So we can get information about relative binding affinity, stoichiometry, enthalpy, and entropy. And this is an example of an interaction, again, between the toxin that we're studying, ricin A chain, and the human ribosomal proteins, ribosomal stalk proteins, showing the traces of this interaction, which also tells us about the mechanism of this binding, and in addition to the stoichiometry. So this is also very useful for studying protein-protein interactions. So we have another subcore, which includes flow cytometry. We have an Accuray C6 flow cytometer from BD, which you will hear about also later on today. So this instrument has two lasers, has a blue and a red laser, and it has forward and side scatter detectors, and it's optimized for detecting those fluorophores that are indicated. In addition, we can use several other fluorophores. So we use this instrument for cell counting. We also look at apoptosis measurements. We study the effects of toxins on mitochondria, so we can look at membrane potential, ROS generation, etc. So it has many uses, and it is being used by faculty on both campuses of Rutgers. In addition, we have real-time PCR instrumentation, which is an ABI Step 1 Plus instrument. And this is a picture of this instrument. And this instrument is used for gene expression analysis, microRNA expression, protein expression, gene detection. And we have developed an assay for one of the toxins that we study, which target a specific sequence in the ribosomal RNA called the sarsen-ricin loop, and depurinate, remove an adenine from this loop. So we can measure the activity of the toxins using this method, and this has been published. And Mike Pierce is teaching a course on real-time PCR with the ABI instruments, and he will tell you more about the course later. So we also have a subcore that consists of high throughput screening instrumentation. We have a RORTE HDA robot from Singer Instruments, and this machine is used for looking at screening yeast deletion libraries for identifying genes for resistance or sensitivity. So this is an example of a screen we did in which we identified mitochondria as a target of this toxin. So it can use high-density yeast or bacterial arrays, and it is very useful for screening libraries, these deletion or overexpression libraries for identifying genes. So in the high-throughput screening core, we also have some microplate readers. One of them is this Biotech Synergy microplate reader, which can do fluorescence, also fluorescence polarization, time-resolved fluorescence, luminescence, and also UV visible absorbance. So we use this. It also has a dual reagent injection system. We use this instrument for high-throughput screening of small molecules using luminescence or fluorescence-based assays. We've been using it for screening of yeast deletion and overexpression libraries, for increased sensitivity or resistance to toxins, for the small molecule inhibitor studies. Also, it's very useful for growth curve analysis. So I just want to go over some of the training we're doing in the core facility. We're offering short courses, and one of those is in BIACORE technology, BIACORE Fundamentals, which is taught by Xiaoping. And we also have help from GE Healthcare in teaching this course. So this is basically co-taught by both places. And we first offered it in May 2011, and also second time in June. And we're planning to offer it again this year in the May-June timeframe. In addition, we teach a real-time PCR course that basically talks about theories and operation of the instrument. So both of these courses are hands-on courses. You actually get to use the instrument and be able to interpret your data. So the real-time PCR was first offered in June 2011, again in 2012, and we'll be offering this again in 2013. So we're also planning new courses. One is with Zeiss, in confocal microscopy, which is planned for, again, May-June 2013 timeframe. And next year, we're planning a course in flow cytometry. So there is institutional support provided for these courses on our campus from the Office of the Director of Research and Dr. Bradley Hillman. So our core facility is very new. It's, you know, as I mentioned, it's established in 2011, so about 2 years old. And we have several users across campuses of different campuses of Rutgers and also UMDNJ. And you see several groups using the confocal microscope, BIA-core, flow cytometry, step OnePlus, and high throughput screening. We have faculty at SEBS, in addition, in the Bush campus of Rutgers, at UMDNJ, Cancer Institute, and both in Newark and Camden campuses of Rutgers and UMDNJ. So it is a campus-wide facility. Some of our publications, so we have been using real-time PCR for some time, so these are some of the publications. In addition, we've been using BIA-core technology since 2004, and we've got a number of publications, and more papers are coming out in this area. We just started using flow cytometry, but like I said, many people are using it, so there will be more publications in this area, and also in high throughput screening. Confocal microscopy, the first paper that was published from Faith Bellinger's group, in fact, made the cover of the journal in 2011. We then had several additional papers using confocal microscopy. So we've been providing letters of support for grant applications of people if they need equipment that is available in our core facility. We've done that for many people for using BIA-core or confocal microscope or any of the other instruments that we have. We also appreciate if you acknowledge our facility, if you have used the facility for the work in your publications. So I'd like to acknowledge the grants that helped establish the facility, which is initially it was established with a grant from NIH for a confocal microscope. We subsequently got additional equipment such as the BIA-core equipment from an NIH UO1 grant. Also, we got the flow cytometry with additional funding from NIH. And NSF helped us with the real-time PCR instrumentation. In addition, Rutgers Academic Excellence Fund helped us with the first surface plasmon resonance instrument, and several different places in the university, such as our dean, Dr. Goodman, Dr. Pazani, Dr. Hillman, and the Rutgers UMDNJ Institutional Support Committee provided support for our core facility. So this is the website of our core facility, and you can read more about these instruments. Thank you.